Hi, today I'm going to show you a promising new idea for how to slow down climate change by preventing nitrous oxide emissions from agriculture. And we're going to discuss this topic across several scales, from the molecular, to the genetic, to the proteins, to the mitochondria of cells, the cellular structure, to the structure of roots and the leaves in the fields of crops, and the global plantings of those crops and their effects on the atmosphere, particularly the greenhouse gases. We've recently published in a peer-reviewed journal, Sustainable Chemistry and Engineering, and a theoretical analysis of what effects these genetically modified plants could have on the control of nitrous oxide and on methane by modifying the proteome of the mitochondria of the plants. So today I'm going to talk about nitrous oxide in particular, because that seems to be the most promising of the two technologies. So we know the context. You wouldn't be reading this unless you thought that climate change is a threat to human civilization. And it is one of the most important threats that face us in the next century. We must achieve a carbon neutral economy, but we also have to eliminate the emissions of other greenhouse gases, such as nitrous oxide. Nitrous oxide is a greenhouse gas that's almost 300 times more potent at producing global warming than is CO2. And nitrous oxide is also the main threat to the ozone layer, which protects people and ecosystems from the deadly effects of ultraviolet radiation. So it's very important that we get nitrous oxide under control. Unfortunately, most of humanity's nitrous oxide emissions come from agricultural soils. We fertilize with nitrogen, either manure in organic farming or synthetic fertilizer, as in most farming. And either one of these methods of adding nitrogen to the soil increases nitrous oxide emissions greatly from those fertilized fields. And in fact, nitrous oxide is the main air pollutant emitted by plant agriculture. Now we've tried different ways of reducing nitrous oxide emissions, but they aren't all that effective. They reduce it by a few tens of percent, maybe 30%, but they also often result in decreased crop yield because they involve decreasing the amount of fertilizer that's added. Now this sets up a terrible conflict between fighting global warming and feeding people. So we're proposing a new genetic engineering technology that can prevent nitrous oxide emissions from crop soils and also remove nitrous oxide directly from the atmosphere. Now, there's only one known reaction that degrades nitrous oxide at standard temperature and pressure here on the surface of the Earth without increasing energy consumption through high temperature and high pressure systems. And there's only one known reaction, and that reaction is catalyzed by the bacterial enzyme nitrous oxide reductase. And that reaction converts nitrous oxide, an important greenhouse gas, into harmless N2. Now, in the soil, the bacteria there can use the nitrous oxide reductase to turn nitrous oxide into harmless N2, but in most agricultural soils, this enzyme is turned off, which saves energy for the bacteria. Now, special bacteria have been developed in the lab that could be that have increased nitrous oxide reductase activity, but when we try to introduce those bacteria into the soil, they rapidly die out, and their effect on nitrous oxide emissions from the soil is limited. Again, about a 30% reduction, perhaps. But more importantly, the bacteria die out. So instead, we propose to introduce the bacterial nitrous oxide reductase, the enzyme, into the mitochondria of plants. Let me explain a bit what mitochondria are. Mitochondria are the powerhouses of plant cells and of our cells as well. Mitochondria and bacteria, they both derive through evolution from a common ancestor. The mitochondria in a membrane space between the outer and inner membranes of the mitochondria is analogous to the bacteria peroplasm, which is between the outer and inner membrane of the bacteria. And in bacteria, that's where nitrous oxide sits and functions. And we think we can introduce nitrous oxide reductase into the inner membrane space of mitochondria. So we're planning to use the plant mitochondria as surrogate bacteria. Now, this has many advantages, and the main one is that the plant root mitochondria live in a protected environment, unlike introducing bacteria into soil environment, which is very competitive and kind of rough out there. There are predators, and there's competition constantly for resources. Instead, inside the plant cells, the mitochondria are protected and supported by the plant metabolism. It's a commensal relationship between the mitochondria and the plant cell. And there are a lot of mitochondria in plant cells. Each plant cell contains about 500 mitochondria. Here you see a cell, a eukaryotic cell, with the nucleus in blue, and all of these little red dots are the mitochondria, and there are hundreds, if not thousands, of these. Typically, plant cells have about 500 mitochondria per cell, and that means that in a square meter of cornfield soil, there are about 10 trillion root mitochondria present. Now, these mitochondria are secure. They're living inside the plant cells. And they're cultivated by the plant cells that provided energy supply, and they produce energy for the plant cells. And so by cultivating these desirable plants that we've modified to degrade nitrous oxide, we can maintain their mitochondria. Now, the mitochondrial proteome can be modified by traditional plant transformation. So we don't need any new technology here. We can use the technologies that we've used for GMO crops for a number of years. And this is because 99% of the proteins that are found in mitochondria are actually coded for on the nuclear chromosome and made outside the mitochondria and then imported in, the protein imported into the mitochondria by mechanisms in the cells that we have in science have described now for the last 2 or 3 decades. Now, these proteins are targeted into the mitochondria by special sequences of polypeptides on the protein, and these sequences act like as if codes of a letter. They're read by the machinery of the cells and directed into the mitochondria through these ports, the TOM and the TIM, and you can look at this later if you wish. So we're going to introduce the nitrous oxide reductase into the inner membrane space of the mitochondria of plants, as well as introducing the helper proteins that are needed. So here we see the structure of the nitrous oxide reductase and a denitrifier. And the actual enzyme is in green and purple here, but importantly, it needs special copper atoms inserted into the enzyme structure in order to convert nitrous oxide into N2. This enzyme, which is called the NAZ proteins, sits in the periplasm of the bacteria, and we're going to introduce it into the inner membrane space of the mitochondria. We're also going to introduce these other proteins that are necessary for providing the copper in their proper states within the protein, and also providing electrons to the process from the electron transport system through these other proteins indicated here by X and R. And all of these will be introduced into their analogous spaces in the mitochondria that are analogous to those in the bacteria. So that's the overall idea of what we want to do to introduce nitrous oxide reductase in plants. Let's assume that we can achieve a functioning nitrous oxide reductase in plant mitochondria. I would like to look ahead now to see whether or not such a biotechnology could have a significant effect on nitrous oxide emissions from crop fields and on the atmospheric levels of nitrous oxide. And in order to calculate that, we have to jump scales from the molecular to the plant, and from the plant to the soil, and from the soil and the field to the global atmosphere. So I'm going to try to do that in the next few slides. So first off, we recognize that we're talking about taking up nitrous oxide from the soil where it's being produced by bacterial action into the roots where it's degraded in the mitochondria to N2. We're also going to be talking about the uptake of nitrous oxide from the global atmosphere into the leaves of the plants, and where it's converted by their mitochondria in the cells of the leaves of the plant into N2. So let's first look at the uptake of nitrous oxide by plant roots transformed with nitrous oxide reductase in their mitochondria. So nitrous oxide is being produced in the soil by the action of bacteria, and we assume that it diffuses through a cylinder of soil around the root and into the root where it is consumed by the action of the nitrous oxide reductase. If we model that cylinder using this equation, which is derived from the calculus for diffusion through a cylinder, then we can come up with this expression that is in terms of all known parameters, like for instance, CB here is the concentration of nitrous oxide in the soil pores during the growth of corn after fertilization. All of these parameters are known, and if we plug those parameters into this equation, we get a potential flux into the transgenic roots calculated to be about 40 kilograms of nitrous oxide per hectare, which is about 4 acres, per year. And that's 8 times the observed emissions of nitrous oxide from fertilized cornfields. In other words, the action of the roots would be enough to completely prevent the emission of nitrous oxide from those fertilized cornfields. And that would prevent, on average, an emission of about 5.5 kilograms of nitrous oxide per hectare per year. In addition, there would be uptake of nitrous oxide from the atmosphere by the leaves. So the leaves have stomata in which CO2 enters the leaf structure and is converted by photosynthesis to fix carbon. Nitrous oxide could also enter those stomata if there was a sink for nitrous oxide in the leaves. And using the mass transfer coefficients and the microbiological parameters for nitrous oxide reductase, we can calculate the flux as this expression. And that amounts, and all of these are known parameters, and if they're plugged in here, we would be able to calculate the nitrous oxide uptake from the atmosphere as to be about 10 kilograms of nitrous oxide per hectare per year. And that's assuming 40 days per year during which the corn is mature. And the total annual nitrous oxide emissions prevention and atmospheric uptake in a fertilized maize field would be about 15 kilograms per hectare annually. So that's quite high. But could this nitrous oxide reductase activity significantly affect atmospheric N2O? Now, we have to recognize that there are natural sinks for nitrous oxide in the global environment, natural sources, natural sinks. And until recently, those natural emissions and sinks were in balance and nitrous oxide levels were low and constant. But then when fertilized agriculture became more and more dominant, the nitrous oxide levels began to increase, and now they're increasing pretty steadily, as you can see by this chart here, where this is the atmospheric concentration of nitrous oxide, and this is the levels per year starting in 1985 and going up to the present day. So a steady increase of nitrous oxide that is equivalent to about an increasing excess of nitrous oxide in the atmosphere of 8 million tons per year in the total atmosphere after the sinks and sources are taken into account. These are actual measurements in the atmosphere. Well, if we transform the global plantings of crops, corn and soybeans and all the other crops with the nitrous oxide reductase, then what effect would that have on nitrous oxide levels in the atmosphere? Well, here we have a model prediction. The red line shows what would happen to nitrous oxide concentrations if the current trends that I've just shown continue. And this shows that from 2020 to 2100, the nitrous oxide levels would exceed 410 parts per million in the atmosphere. But if 32% of all global plantings were transformed with nitrous oxides reductase starting in 2030, then nitrous oxide concentrations in the atmosphere can be stopped. Their increase can be halted by the addition of this new sink for nitrous oxide in plant roots and leaves. And if 72% of all of the global plantings of agriculture were transformed, then we could return nitrous oxide concentrations in the atmosphere back to their pre-industrial levels by the end of the century. And even more removal of nitrous oxide if 100% were removed, were transformed by nitrous oxide reductase. So can nitrous oxide reductase expressing plants, can they slow down the rate of climate change? CO2 is the main driver of climate change, of course, and it's currently increasing in the atmosphere by about 2 parts per million by volume per year, or about 20 billion tons of carbon dioxide per year in the total atmosphere. If all crops were transformed with nitrous oxide reductase, then 11 billion tons of CO2 equivalents per year could potentially be prevented and removed from the atmosphere. So this is assuming 100%. This is assuming 100% of the crops transformed, and that would reduce nitrous oxide very rapidly. And if that were done, and considering the fact that nitrous oxide is 300 times more powerful than CO2 as a greenhouse gas, then the equivalent of 11 billion tons of CO2 equivalents per year could potentially be prevented. And that compares to 20 billion tons of CO2 being produced in excess every year. So in other words, nitrous oxide reductase technology, if widely applied, and if it were successful, could significantly slow the annual rate of increase in global warming. Nevertheless, the CO2 would continue to increase, and unless we move to a zero carbon economy and we remove CO2 from the atmosphere, then atmospheric CO2 will continue to increase and will eventually overwhelm the effects of removing nitrous oxide, and we'll be back to the situation of global climate change again. But this will buy us some time. Importantly, the cost of this technology would be low. Once the nitrous oxide expressing plants are created, seed production would be the same cost as seed for non-transgenic plants, and the production could be scaled up with no additional costs. We know how to scale up seed production better than anything else. With the current carbon prices of about $15 per ton, that farmers would be able to claim carbon credits on the carbon market, which are currently equivalent to about $47 per hectare, or about almost $10,000 per year for a typical US farm growing corn. So in conclusion, we think that the proper location for nitrous oxide reductase enzyme in engineered plants is in the mitochondria. And we think we can construct these complicated molecular machines in the mitochondria because they are analogous to bacteria. And this technology, we calculate that this technology of nitrous oxide reductase in plants could potentially prevent any crop field emissions of nitrous oxide and directly remove nitrous oxide from the atmosphere on a global scale. This technology by itself, sustainably and at little cost, could potentially stop increases of atmospheric nitrous oxide. The widespread application of nitrous oxide engineered crops could significantly slow global warming. The technical challenges of successfully doing this, of expressing nitrous oxide reductase in plants, those technical challenges are high, are great, but the potential payoff are large enough to be worth the risks. And we think that it should be doable because we have in the past expressed simpler systems in plants successfully for the degradation of pollutants. This system is more complicated, but it is within the scope of genetic engineering technology. Well, if you like these ideas and want to help us make them work, please click like below and leave a comment, and visit our lab website, which is given here. And if you wish, click the donations button. While you're there, you can also learn about our genetically engineered houseplants to help clean your home's air. Here are some of the changes that we've made since our initial YouTube presentation. We've tried to improve our calculation. The latest one down here, number 6, is that we've reduced the effective season length by 50% in order to account for the growth period and the lack of full maturity earlier in the season.